I'm David Versluis from Leiden University, and with my collaborators, we investigated how and why specific bacteria may grow in the newborn gut using a computational multiscale model of the infant gut microbiota. We know that in newborns, the gut is typically first colonized by oxygen-using bacteria, like Enterobacteriaceae, and a few days or weeks later, anaerobic bacteria, like Bifidobacterium, typically become dominant. But this varies across infants and influences the health of the infant. There's a hypothesis about this change being related to depletion of an initially present volume of oxygen, with no actual models for why these species in particular dominate at these times. That is something our model can help explain. The model works by representing the colon as a grid and populations as species-specific genome-scale metabolic models. We calculate metabolism and growth for each population and integrate that over the whole gut over 3 weeks of time. This allows us to model variation between infants and complex processes in the microbiota, like cross-feeding, that are so important in this system. Here, you see the metabolites taken up and produced by the major bacteria at one time point of the system. As you can see, bacteria also cross-feed. That is, they feed on metabolites produced by other bacteria. This leads to a very complex metabolic composition. One of the things we do with the model is that we take a look at both a situation without initial oxygen on the left and one with on the right. Without oxygen, we see that of our 15 species, Bifidobacterium becomes dominant nearly immediately. But when we do add oxygen on the right, we see that while it's depleted very quickly, it gives E. coli and Enterobacteriaceae such a boost that it continues to be the largest species even after the oxygen is depleted, until it is often surpassed by Bifidobacterium, which is exactly what we see happen in infants. In this way, the computational model allows us to generate hypotheses on bacterial colonization and succession in the infant colon.